Good morning. I'm glad to be here and to speak about multiplicity corrections in bioequivalence trials. Today morning I heard the word bioequivalence already twice, so it seems that this is an interesting topic also in these type of trials. Next slide, please. Bioequivalence trials are important in the drug development process. In the innovative pharma industry, they may be used, for example, to bridge the clinical phase 3 formulation with the final to be marketed formulation, or to study drug-drug or food-drug interactions. Obviously, for generic pharma industry, the results of bioequivalence trials are the pivotal evidence that must be provided in order to obtain marketing authorization. Next slide, please. Two products are bioequivalent if their bioavailabilities, namely the rate and extent of absorption, lie within predefined limits, acceptable limits. These limits are set to ensure comparable in vivo performance in terms of safety and efficacy. The majority of bioequivalence trials are comparative pharmacokinetic trials, and therefore the rate and extent of absorption may be described by two major PK metrics. There are Cmax, the maximum plasma concentration that reflects the rate, and the area under the curve that reflects the extent of absorption. Next slide, please. The gold standard in bioequivalence testing is the randomized single-dose, two-period, two-sequence crossover design when comparing two formulations. The guideline says that there are alternative design, and under certain circumstances, provided that the study design and statistical analysis is scientifically sound, the alternative design, well-established design, could be considered. And these designs are, for example, parallel design, replicate crossover designing that are useful for highly variable drugs, or sequential crossover design. But frequently in drug development, we need to conduct study with more than two formulations, and this possibility of running crossover design with more than two formulations is actually not explicitly mentioned in the BE guideline. And I believe that these trials, they do fulfill the guideline requirements on alternative design. And I have an example here, actually, please. Next slide. So imagine a situation where 4 formulations, A, B, C, and D, would be compared to reference product E. The study could be done in Williams design, but also simple Latin square would work as well. And the study statistical analysis would be done in accordance with the guideline investigation of bioequivalence, namely with ANOVA, and the bioequivalence would be assessed by means of confidence intervals. Next slide, please. When speaking about the study design, we have to keep in mind that a clear objective of the study must be pre-specified, and obviously the equivalence margin has to be pre-specified in the protocol. So in our example for the 5-way study, the objective would be to compare the rate and extent of absorption, and provided that the results are conclusive, they would be used as a proof of bioequivalence. Next slide, please. And now I'm coming to the statistical analysis, and basically we have 3 possible scenarios or situations. In the situation 1, that is so-called joint decision rule, there is no alpha adjustment needed, because the aim of the study is to show that all test formulations are bioequivalent to the reference. But frequently, we want to have the option to choose either one of the bioequivalent test formulation, and that, of course, needs alpha adjustment. We would need to adjust the family-wise type 1 error. There are a number of procedures, and the most well-known is the Bonferroni, but there are also other procedures, like procedures of Holm and Hochberg, that are more powerful. There is one procedure which we actually initially considered, that is the procedure that is suitable for the situation many-to-one comparisons. That is a Dunnett procedure. However, this procedure is not compatible with the requirement of guideline that says that each comparison, each ANOVA analysis must be done separately for each comparison. I'll come in one of the next slides. I'll come back to that. Next slide, please. So there is also situation number 3 that has been discussed in London during the QA symposium before the new BE guideline came into force. And the situation 3 is a situation where we do have one lead formulation and the other formulations are included in the study only for exploratory purposes. So obviously for this type of trial we don't need the alpha correction. And interesting is that already in 2010 the possibility to run a trial with alternate lead formulation has been mentioned, that correctly also was mentioned that this is a situation where alpha adjustment comes into play. However, still the agencies somehow feel that it's more appropriate to run 2 separate studies, which I don't think it's needed. Next slide, please. And this is the guideline requirement that says that in studies with more than 2 treatment arms, we need to run separate analysis. So we need to exclude the data from the treatments that are not relevant for the comparison in question. That practically means that we have to calculate separate ANOVA analysis and apply the correction for each of the analyses. And I would like to show you in the next slides, I would like to show you the performance of the 3 procedures, Bonferroni, Hofmann, Hochberg, and in terms of the confidence intervals. And next slide, please. And so to illustrate the performance, we actually reanalyzed a study, the 5-period study, where 20 healthy volunteers were enrolled, and we, as I said, we applied the corrections for in the ANOVA for each of the comparison. And the results are depicted on the next slide. And I plotted here the 90% confidence intervals around the test to reference geometric mean ratio for each formulation A, B, C, and D. And so what is obvious? Obvious is that the procedure of Hofmann-Hochberg, there the confidence intervals are narrower, and it's been previously shown that these procedures are more powerful than the procedures of Bonferroni. And you might have now realized that some of these confidence intervals are not symmetric around the test to reference geometric mean ratio, and this can be explained by the fact that the adjustment for the lower and the upper t statistics in the 2 one-sided t-test is done separately for lower end and for the upper t statistics. So to sum up at this point, next slide, please. I would say that there are some procedures that would be the Dunnett procedure, for example, that cannot be used based on the guideline requirement. However, we can choose other procedures that are compatible, and here we would recommend the procedure of Hofmann-Hochberg, since they've been shown previously as being more powerful than Bonferroni procedure. And there is little published in the area of bioequivalence trials, and even less is in the guideline. So we would be really happy if our discussion would somehow trigger some changes in the guideline. Thank you very much.
Thank you for a very interesting presentation. And now we invite Thomas Lang.
Hello and good morning. I'm looking forward to discuss this presentation. It's a very interesting topic. I'm not sure whether we need that much of changes in guidance documents. I don't think that the current wording in guidance or in related guidance documents is in the way of such an approach in general. But let me just repeat the question with the first slide. Which statistical procedures to preserve the overall type 1 error could be compatible if we have to analyze bioequivalence data in separate ANOVA models. Let me, with the next slide, just provide you with some, with one context-related comment, and I think the colleagues who developed the update of the bioequivalence guideline did not have your specific scenario in mind when they decided to put down something that separate analysis need to be conducted as soon as more treatment modalities than one are involved in a bioequivalence trial. What they had in mind, obviously, was 2 or more reference compounds where there was one test drug to be investigated. These 2 or more reference compounds being or coming from different regions. And the other situation might have been that you have completely different treatment modalities, for instance, fasting and fat conditions, where you also classically would have we have to meet all the hypothesis situation. So this is just for the background. And I'm not in the position for the next step, please. Of course, not in the position to judge the need or the frequency of this particular situation in the development of a generic, where it is very important to have more than 2 test formulations in one trial to be compared to one reference. So I'm not in the position to judge this, but it was very interesting to think about it. And my conclusion was that actually the recommendation as it is in the guidance document for separate analysis would not change in this regard, because as far as I followed all the discussions in the past, regulators, and there is a tight setting of a very standardized setting, of course, for bioequivalence, do not want the estimates of the pairwise comparison of interest to be affected by any other parameter that might be avoidable, and such a parameter could be a treatment factor from from more than 2 treatments. This means that the mean square error that usually results from a full data model where you have all the all the factors, all the treatments in the model, would not fulfill this criterion. So I think there is little room for other alternatives, as other discussions around bioequivalence are that detailed and sophisticated that any other situation that might allow several approaches for the analysis to be conducted, I see no possibility for that. So next slide, please. Back to the question. And the next, please, just to set up a more formal background of the problem that was just stated. If we have K treatments or K pairwise comparisons, K hypotheses, and according to my understanding, in the simplest way, there would be no hierarchy for these pairwise comparisons and for these hypotheses. And the goal of such a trial would be to have at least one of the test formulations to be bioequivalent to the single reference compound. And one meaningful requirement would here also in this case be to control the family-wise 1 error in the strong sense. Next slide, please. So the answer to your question would be that in principle, if such a trial would be set up and if such a set of hypotheses would be tested, any multiple comparison procedure based on p-values coming from these k-separate tests, from these k-separate ANOVA models, controlling the family-wise rate in the strong sense might finally be acceptable. And that's the reason why I said in the beginning, I don't know whether we have a very different situation here compared to a, let's say, phase 3 setting where we look at 2 or 3 potential endpoints that finally one of them needs to be shown to be significant. So for me, I don't see a big difference to this situation. And what does this mean in practice? Of course, the proposals brought up by you. Next slide, please. Of course, when it comes to the to the testing situation. Bonferroni correction could be done in general, or any sequential or any other sequentially rejective method, e.g. the Holm method, also the Hochberg method, if all the requirements can be shown to be fulfilled, can be used from my perspective. And as long as this condition holds that these hypotheses are of equal relative importance, it does not seem that more sophisticated approaches would be required for that purpose. But for other scenarios, even these could be considered, I think. But from our perspective, from our regulatory point of view, we haven't been confronted with such proposals in the past. And yeah, we would need a look at it against this background I just presented. Just the last section of this slide, please. You mentioned the publication from Zheng, Wang, and Zhao, who just did a simulation study and evaluated the also power properties for several sequential rejective methods under this scenario. But what they did, according to my understanding, was also making use of the full data model. So they just took the mean square error that resulted from the full data analysis, using all the data from all test treatment conditions at the same time in one analysis of variance. And they resulted in a recommendation that the Hochberg procedure might be preferred in this situation. Final comments. Of course, and the paper just mentioned here reflects this, we are in a situation where we have a two endpoint situation, which means that if we are talking about optimality of a specific multiplicity correction method, one might be interested also in the correlation between Cmax and AOC parameters. And one of my questions was whether optimality in terms of type 2 error might also be dependent on the design of the bioequivalence trial in general. And the last question, or could be the most provocative question, is how would asymmetric confidence intervals resulting from, for instance, Holm and Hochberg, be perceived in the community when it comes to making the decision of bioequivalence, not on the basis of the test result, but on basis of the confidence intervals as they are usually, as it is usually done. Although there might be this one-to-one correspondence, but we always have also to think about the perception in the community in general when such proposals are made. Thank you.
Thank you, Thomas. And a comment from Jiri?
First of all, I agree with you that the problem of common variance must be addressed, and I'm not disputing that. We can imagine a situation where products having high variability could somehow damage a product with lower variability, or vice versa, where a product with low intrasubject variability would make the estimate of variability lower. This is just how it works with the common variance. So I agree with you on that. It's there is the need to separate the ANOVAs, and I'm also glad that you mentioned that that procedures multiplicity adjustment would be possible in this situation. Of course, the guideline cannot cover all the situations, and then industry is always happy to come with something that doesn't fit the guideline specification. But this strategy is quite common in the industry, at least in the generic industry, and the possibility of using such a study as a pivotal evidence would, of course, bring efficiency to the drug development, and this has been mentioned today in the morning. And I believe if it's done correctly, why not? And to particularly interesting question is how the asymmetric confidence intervals would be perceived in the community. Well, I don't know, but typically for bioequivalence testing, we've got two one-sided t-tests and the corresponding 1 minus 2 times alpha times 100 confidence interval is equivalent to those two one-sided tests. So, as you mentioned, it's should be actually the same.
Okay, thank you. Thomas?
This is not a direct comment back, but a general remark from the history of assessments we did at the agency. Of course, we are often in a situation where we do not have, when it comes to generic applications, we are sometimes in a situation where we face the very problematic situation where we have one positive bioequivalence trial and perhaps a history of failed trials with compounds that are similar but with a kind of modifications from the manufacturing side or whatsoever, from early development batches where we see negative results. And actually, under such a scenario, we have been frequently in a situation where no one cared about the type 1 error in the past. So we had this whole bunch or this whole set of trials, and the assessment was on basis of all these results that had been presented. So from this perspective, regulators should be open to a more, if a generic company has many formulations or two formulations at the same time and is not aware of the chances of being bioequivalent to the originator. This, compared to the situation I have just described of sequential trial and error, might be an alternative which we should think of. It's not too bad from my perspective, if we have a prospective way of thinking about the type 1 error here.
Okay, thank you. Any comments from the floor? Yes, Martin?
Maybe first two technical comments. I think one, what was already Thomas brought up is the confidence intervals. I think one in general has to be very cautious in constructing confidence intervals for sequentially rejective multiple testing procedures like the Bonferroni-Holm or Hochberg procedure. It's typically, if you just use the significance levels that you get when performing the test, this will not result in simultaneous confidence intervals then for each of the parameters. So the one has to be really cautious, and there's some literature on how to construct really simultaneous confidence intervals in this setting. The other thing about the Dunnett test, and the improvement the Dunnett test brings over the Bonferroni test, that it exploits the correlation between the test statistics, because we do the comparison always to the same control group. And of course, one could consider also other statistical methods that somehow take into account this correlation when determining the critical value, still satisfying the requirement of the guideline that you do separate analysis for each of the comparisons. And maybe the final point, which was already also raised by Thomas, the question on if separate studies should be performed or several compounds compared in a single study. I completely agree with Thomas that if you perform separate studies, of course, you have no control of the type 1 error rate over the several studies performed. Even worse, the statistics will be independent, so the chance of making at least one type 1 error is larger than even if you would do no multiplicity correction, but estimate the equivalence in a single trial. So from this respect, the approach to investigate them in a single trial is in any case preferable. And in addition, you have the possibility also to compare the agents between each other, since you have randomized them simultaneously in the same trial.
Okay, thank you. Jiri, would you like to?
Well, why it should be preferable to run two studies one after one. I mean, if you are able to show bioequivalence with properly adjusted alpha in one trial, what would be the benefit of repeating the trial. So frequently, bioequivalence or companies, generic companies, run a study with 4 formulations, and they have to repeat the study even if one of the test product is bioequivalent. For me, this is wasting of energy, wasting of time, and maybe also ethical issue.
David, please.
Or just a comment. Obviously, I think traditionally generic companies they invest a lot of effort in the pharmaceutical angle of developing the generic product. And so they're not just trying formulations, you're investing. So when you come to do that PK trial, you're fairly confident that you're going to be bioequivalent. Sometimes you're not, and you investigate that and find reasons why you need to adapt the formulation. So whilst I accept from a methodological point of view, putting in a whole basket of formulations is attractive and can control the type 1 error, if that meant you hadn't fully investigated the pharmaceuticals of the product first, which I would imagine might be attractive in terms of saving money, then that would be a retrograde step in my mind. So I like the sequential approach if you're learning as you're going along. But so I'd be slightly cautious about putting, for example, half a dozen formulations into a bioequivalence trial, correctly controlling the type 1 error and then concluding that bioequivalence had been shown.